Hello everyone, my name is Beril and I'm a PhD student at EPFL, and this is a joint work with Yinan, Zhang and Pascal Frossard. Thanks to the recent advances, multiplex imaging allows for easier multi-label analysis of tissue biopsy samples, enabling the study of complex spatial biology within the tumor and the tumor microenvironment. However, very often, with this technology, the biopsies are scanned first at a very low spatial resolution, and guided by experts, some region of interests are scanned for a second time at a higher resolution that gives us the opportunity to do cell-level analysis. Although very promising, this technology is relatively new, and labeled or even unlabeled datasets are very limited for data-driven approaches. On the other hand, it is now widely accepted that the human microenvironment carry valuable information for cancer prognosis and personalized medicine. The analysis based on spatial organization of different types of cells, as well as the interaction between them, is a very active research topic. Once segmented, we think that the inherent non-grid structure of cell locations makes graphs a perfect fit for such analysis. Defining each cell as a node based on cell location and constructing the graph based on spatial distances is a good start point for using tools from network analysis for biomedical images. In this work, we focus on a scenario where few slides are available with no associated labels, and every slide is described by cell-level analysis of region of interests extracted from different parts of the slide, which means we have access to cell locations and phenotypes. In the proposed pipeline, we first construct the cell graphs for each region of interest, and starting from cell phenotypes as one hot encoded feature for each cell, we obtain an embedding for each graph using graph neural networks. In particular, graph stage layers followed by differentiable pooling in order to take multi-hop interactions into account. Once all regions are associated with an embedding, at the first stage of training, we use a permutation invariant aggregation function to obtain the slide level embedding, which is later used for a self-supervised classification task, namely patient or slide identification. In order to incorporate interpretability, we employ a differentiable graph sampling module, which selects a subset of a region of interest to obtain the slide embedding. By using different learning rates for pre-trained network and sampling, we realize the second stage of training for the same self-supervised task. For testing the proposed pipeline, we generate a synthetic dataset with 10 different patients, or namely 10 different whole slides. Each slide is assumed to have 15 different regions of interests, each of which refers to a different cell graph of a fixed size. Each cell graph can contain up to 6 different cell types, imitating tumors, stroma, and different types of lymphocytes. We consider tumor stroma border, known as invasive margin, and define 6 different distribution of graphs based on the combination of present cell types as well as their spatial distribution. The graph 0, representing the human desert with no lymphocytes, is considered to be non-discriminative and is present for all the patients. Each patient is further described by a combination of 2 types of discriminative patches, which you can see in this table. And our purpose is to be able to identify patients by jointly sampling the discriminative patches associated with them. For the learning scheme, for each patient, we split patches into 3 equal parts for training, validation, and testing by keeping the distribution of discriminative graphs similar for each subset. At the end of first stage of training, we can reach to 100% test takers for a patient classification task for the story example. After training with the graph sampling module, we test by realizing 100 Monte Carlo simulations for each of which we sample k different graphs for each patient. If we sample 2 graphs, as you can see here, the accuracy slightly drops to 80%, while we are able to sample both of patient-specific discriminative graphs half of the time, and at the other half, we can sample at least 1 patch correctly. If we increase k at that test time, we observe a slight improvement for graph sampling performance, as one might expect. As a result, we here provide a proof of concept performance evaluation for our novel pipeline for pre-training multi-graph systems on a particular application of biomedical image analysis. We think that this work can bring insights for different problems where the dataset and labels are limited, and the methods can profit from pre-training to be later transferred for any supervised task. Thanks for listening. And please do not hesitate to drop us an email if you have further questions.